Hello, my name is Jennifer Fish. I'm an assistant professor at the University of Massachusetts Lowell, and in my lab, we investigate disease mechanisms of SATB2 associated syndrome. So today I'm going to give a brief overview of this syndrome and some of the work that we're doing in my lab to understand the mechanisms of disease and what we might be able to do in terms of therapies. So what is SATB2 associated syndrome, also typically referred to as SAS? So SAS is a genetic disorder that results from the mutation, which can also be a deletion, of a single allele within the SATB2 gene locus. So this syndrome has now been well characterized. It has an onset before the age of 2 with most of the syndromes. In fact, many of children are born with cleft palate, so that would be at birth. And some of the disease symptoms or phenotypes that are associated with the syndrome are intellectual disability or developmental delay, severe speech disorder. They have craniofacial and dental abnormalities, some metabolic abnormalities and behavioral issues, and also an overall bone, sort of brittle bone is often associated with this syndrome as well. So a major question that we are trying to address in my lab is what is or are the disease mechanisms behind SAS, because we know that SATB2 is expressed in many different cell types and that what it does in each cell type is slightly different. So we address this question in a cell type specific manner to understand the disease mechanism. And then we secondly are looking to address therapeutic approaches. So at the moment, a major avenue that we are using to address these questions is induced pluripotent stem cells. And this has been possible through the SATB2 Foundation, which has supported the generation of these cell types. So what is an iPSC? So the way these cells were generated is that some tissue is taken from a human individual. And in this case, we had the patients as well as one or both parents. And in the case of the SATB2 Foundation cells, we took fibroblasts. So just a small sample of skin, typically from the arm. And those cells are then put on a dish with some liquid we call media that gives them the nutrients they need to grow. And those cells can then be reprogrammed. And so what that means is that we overexpress certain proteins to make these cells be more like embryonic stem cells. So what typically happens in development is that cells turn on genes that they need to do certain things. For example, heart cells turn on heart genes, muscle cells turn on muscle genes. And that gets sort of integrated into the DNA. So this reprogramming takes away those marks on the DNA that tells a cell it's a certain type so that it can now be differentiated into whatever sort of cell type we want it to be. So we can take skin cells from an individual, reprogram them, and then differentiate those cells to make either neurons or bone cells, which are the major cell types that SATB2 is expressed in. And then we can study disease mechanisms in those cell types. So what does that look like? So on the left side here, you see these are the fibroblasts. So when we got this piece of skin, what we did was we placed it on the Petri dish, and then the cells grew out. They migrated away from that piece of skin and covered the dish. And then with the growth medium, they were able to proliferate and make more cells. Those were then reprogrammed to generate these induced pluripotent stem cells. And that is what you can see here on the right. So these induced pluripotent stem cells form these colonies. So this is what, this is not just one cell, this is a bunch of cells. It's really hard to see them, but they're all forming this distinct colony. Now these cells, if we culture them or we put them on a dish, as I said before, we have this media that has the nutrients. Different cell types want different nutrients. So we can put on that dish the specific nutrients that those cells need to make neurons or to make a mesenchymal stem cell, which would give us bone, for example, osteoblasts. And that's how we then get these different cell types that we can study. But for the moment, we have been trying to do some initial characterization in these induced pluripotent stem cells. And so just a little bit of a background is that on this SATB2 gene and the cells that we have is shown here. So we are currently working with 3 different patient cell lines that are identified by numbers, different numbers. And so we have 3 different types of mutations that we are studying. So the ID100 is a missense mutation. So that is shown here. So it's at the end of exon 7. A missense mutation means that the amino acid that is supposed to be in that location of the protein, so a protein is just a big chain of a bunch of amino acids, the missense mutation changes the identity of a particular amino acid. And so there's just one wrong amino acid, and that causes a problem. We also have the ID59. So this is a nonsense mutation. And what a nonsense mutation is that when that mutation changes, it makes a stop codon. And so the stop codon is telling the cell that that's the end of the protein. So this is a premature stop codon, so it makes a short protein. And that's also a problem because you need the whole protein to do its job properly. And then we have this ID2, which is a deletion. So that's just that part of the DNA is missing. So of course there's no corresponding amino acids. So for each of these individuals, this mutation is on one allele, so that they will make one normal protein that is from the healthy allele, and then they make one protein that is either missing in the case of ID2, has one wrong amino acid in the case of a missense mutation, or is too short in the case of a nonsense mutation. So one of the things that we're really interested in knowing is what is the effect of these mutations on the levels of mRNA and protein, because that's what the cell is going to use to perform the function of this protein. So to address that question, we have quantified the levels of SATB2 mRNA and protein in these induced pluripotent stem cells. And the way that we can do that is, so to quantify the mRNA, we extract RNA from these cells, and then we use what is called primers, are these small pieces of artificial DNA, if you will, that match or bind to the SATB2 gene. So then they can identify the SATB2 gene, and we use a sequencing technology to quantify the number of these little mRNA molecules. So we can count them relative to a healthy individual, so someone that has 2 normal copies. And then the protein we identify through using a Western blot. So we can, in this case, we use another tag, an antibody, that helps us identify that protein, and then we can quantify it just visually. So in the case of looking at mRNA, we have 3 individuals. So in all of our controls we've used for this initial experiment, the maternal, so that's what the M is for. And we quantified the amount of RNA in one of these individuals. So in this case, we took MID100. And then we said how much RNA, SATB2 mRNA, is in the other cells relative to this one individual. So that is why we have this arbitrary 1. So this individual was set to 100%, and everything is relative to that. And you can see that even among the maternal individuals, and the different colors just refer to 3 different quantifications that we did, that there's some variation among those 3. So this parental individual has less SATB2 than this one. But it's not, this looks like a big difference, but it's actually not in this type of quantification. What is interesting is the individual number 2 that has the deletion. You can see that there is quite a difference and we get below 40% on some of these. So these ones are a little bit lower. And this is what we would expect because there's a deletion, there's no RNA produced because that piece of DNA is missing. With these other two patients, you can see that they are pretty similar. They're not much lower than these maternal individuals. And that's also to be expected because there are mutations there, but there's still the RNA there. So this technique is not able to distinguish between an RNA that has a mutation and an RNA that doesn't. It just knows is there RNA. And so that's what we are able to see. We're able to detect a little bit that there's reduced RNA in this individual that has a deletion, but the ones that have a nonsense and point mutation, they look very similar to the parents. So the next experiment that we did was to look for a protein. And in this case, what we do is we extract the protein from a cell and we run that on a gel. And we use an antibody that is going to detect the SATB2 protein. And that antibody has a fluorescent tag on it so that we can then see it. We have a way to visualize it. And so it's going to bind to our protein of interest, and then we can see what's bound because it has a tag. And what is shown here is we've run the protein from the 3 patients, ID2, 59, and 100, and 2 of the parents, 59 and 100. At the time, we didn't have the MID2. And the SATB2 band is this one. And what I hope that you can see is that it's very small or light. It's not dark, and it's not very different among any of the patients and the parents. And that is relative. So this is what we use as a positive control. This last lane, which is SATB2 in bone cells, which you can see is a quite bright band. And this is a cell line where we know SATB2 is highly expressed and important to function. So what we interpret from these data is that SATB2 in these pluripotent stem cells is not very highly expressed, and we're not going to be able to detect the differences that we're looking for. And this also is not unexpected because if SATB2 were super important to these cell types, there would probably be no development. So the fertilized embryo would probably not develop, and we wouldn't see individuals with SAS. So what we're doing is that we are going to differentiate these iPSCs into mesenchymal stem cells. So these are these bone type progenitors where we know SATB2 is highly expressed, and then repeat these experiments. And we expect in this cell type that we will be able to see differences in protein levels between the patient and the parent. One of the things that we're working on is trying to develop another antibody that will help us with this assessment. And to go back here and explain why that's important. So we have an antibody that detects the C-terminal. So the C-terminal is the end of the protein. And so that's where the antibody binds to that part of the protein and allows us to see it. Well, if there is a nonsense mutation and the protein doesn't have that last part, then the antibody won't be able to detect it, and we won't see if this mutated protein is being produced. So we need an antibody that binds to this N-terminal region. There are some on the market. We haven't found them to be successful in identifying the SATB2 like we see here with the C-terminal. So we're still working on that. So what we are doing at the moment is differentiating these cells into bone precursor cells, and then we are going to assess the production of these deleterious mutations. Further, we are going to be testing a series of drugs. We have received funding also through the SATB2 Foundation and through the Arkansas Medical School to use drugs that will allow us to read through the nonsense mutation. So what that would do is that instead of prematurely stopping, it would read through that stop codon and make the whole protein. So we would be able to detect if those drugs are working in these cells. So those are ongoing experiments. Additionally, we are planning to differentiate these iPSCs into neurons. And in that case, what we need to observe is what is the effect of the mutation. And we want to do that, so the characterization of cellular phenotypes, so that we can further use drug screening. So there are many, many available known therapeutic drugs that may potentially ameliorate some of the symptoms of SAS. But in order to know that those drugs are working, if we apply them to the cells, we have to know what is wrong. We have to have a sign that we can observe. So, for example, these are images. So there are 6 different boxes here, and these are 6 different cell types. The one here in the top left is a normal, healthy bone cell. And these other ones, with the exception of the C20, are cells that have mutations in SATB2. And you can see by these asterisks that there's some weird shapes. So this is the DNA, has these different weird shapes, and we get really strangely shaped DNA. Sometimes we have these, what we call blebs. So these are what we refer to as phenotypes. So this is, the mutation is causing some abnormality in the DNA. And this is the type of thing that we want to identify in the neurons, so that then when we apply the drug, we can see if that abnormality goes away. So that's what we plan to do with the neurons. And then finally, we're also currently working with the mouse model, and we are trying to make a complete characterization of SATB2 expression in all mutant phenotypes in this mouse. So that includes the postnatal expression, particularly in different aspects of the brain, and other things that might be contributing to metabolic disorders, as well as how these SATB2 mutations might be affecting structures such as the jaw muscles and innervation that might be contributing to some of the other phenotypes that haven't been as fully described as the bone and brain phenotypes. And finally, I would just like to acknowledge the people in my lab. This is our COVID era photograph of everyone with their masks on. And at the moment, there are 2 individuals that are working really heavily with the iPSCs. That's Marcelo Costo and the postdoc in my lab, Evelyn Schwager. And then we also have Annalise Lorace and Hui Win, who are working on the mouse bone cells on the SATB2 project. So most of the people in the lab, and actually Arusha also is working on the mouse model. So basically everyone but Nate is working on SATB2. So thank you very much. And we would appreciate further questions. If you have them, please feel free to contact us or the SATB2 Foundation. Thank you.